I'm Michael Overduin, and I've been here in Birmingham for almost 10 years now. I came from the US, and I came here to build a facility where we have large magnets to look at very small molecules, trying to figure out how life operates in terms of the molecules that are in our cells. We put them in a test tube and look at each atom and see how they form shapes, three-dimensional shapes, and interact, communicate with other molecules we put in the test tube. So we look at the form and the function of molecules in a solution that's a lot like what you find in a cell. We try and find out how proteins behave. And when we understand how they behave, we can figure out how to change their behavior by designing small molecules, inhibitors, for example, or drug-like molecules, to see if we can change their activity and produce a more healthy response. We're trying to figure out how the molecules of life work, and often these proteins are mutated. For example, in a tumor cell, you'll have mutations that alter protein structure. And so if we can see how those mutations affect protein structure and how they affect how proteins communicate with each other, we then have a way into designing new therapeutic agents. We can try and find a small molecule to jam a misbehaving pocket, make the protein behave differently. And so a lot of what we're trying to do is provide the information that drives drug discovery. What really fascinates me is the organization in the cell. We know that DNA encodes RNA, and that produces proteins. And what I'm interested in finding out about is how proteins create cellular structure, how proteins get to a particular destination in a cell, the Golgi apparatus, the endosome, the cell membrane. There are specific signals that send proteins to a destination that drive them home. We want to figure out those signals, the chemistry that brings a protein to a membrane and changes membrane structure. When we do that, we can understand how cells are formed. And of course, we're walking collections of cells. And so we can figure out how cells are created and how they misbehave, how they move inappropriately in a tumor, for example, that becomes invasive. Then we have really useful information, again, for trying to design new therapeutic agents. And so we're much more complex. And so it's challenging to discover molecules that will penetrate into our bodies, find a specific cell type, and then find a protein within that cell to switch that protein off. And I think by working together with drug companies, with small companies that have really great technologies that they can offer, and also universities where you have this wealth of knowledge, if you can bring all that together, you can really start to make a difference. If you have too much of this protein, the breast cancer cells become invasive, they spread. And so we're trying to figure out how this kinase works that turns on this invasive behavior. And if we can figure that out, we can find hopefully a way to jam the system so that the cells don't become invasive. So I've had a student in my lab working on the structure of this protein, this kinase, trying to figure out the switch that can turn it off and on. And we found that we can see the structures of both those states, and now we can try and find ways to design small molecules to correct the problem. I'm really fascinated in how biology works. I want to see how molecules create organization, not just fixed organization, but dynamic organizations, like moving cells, moving organelles, molecules that move around the cell. I want to know how that order is created. We're organic and messy individuals, perhaps, but beneath that, there's a code that says exactly what kind of cell type, what kind of cells that you'll be able to form as throughout your development. There's a huge amount of program in our DNA. And so trying to read that DNA program, trying to figure out how that, in the end, creates cellular structure is a huge intellectual challenge that we want to chip away at and really figure out some of the codes that create that order in a cell from that original information in the DNA. So we're, I would say, not even halfway there, but we're getting there slowly but surely. And I think there's now so much information now that we know the genes that make us who we are, and we know the proteins they encode. We can start now to chip away at how that creates order, that creates cells and tissues, and hopefully we can make real progress in the next decade in terms of using that to be able to develop new therapeutic agents. So we're getting there.